Hi, my name is Sepideh Sadegh, and I'm a PhD student at Technical University of Munich. Today, I'm going to present NeDRex, our network medicine platform for drug repurposing. Drug repurposing or drug repositioning is the process of identifying alternative uses for existing approved drugs. Compared to traditional drug development, drug repurposing has advantages such as lower cost, reduced risk, and faster drug development timelines. Because most reposition drugs have already passed the early phases of development and clinical testing, they can potentially win approval in less than half the time and at quarter of the cost. Drug repurposing can begin with target discovery for an existing drug, directly followed by phase 2 and 3 clinical trials, while animal and phase 1 clinical studies were not conducted as results for these studies are already available for an existing drug. There are various ways of categorizing computational or in silico drug repurposing approaches, and mainly there is not a clear-cut distinctions between different categories. To name a few, there are signature-based approaches where gene signature information obtained from disease omics data is used to discover new off-targets or mechanisms of disease. We also have similarity-based approaches where it's assumed similar drugs are indicated for similar diseases. Another class of approaches that has been increasingly gaining attention is network-based approaches. These approaches can leverage many different types of information and aren't limited to one single repurposing strategy. For example, pathway mapping approach from this figure can be considered a network-based approach as well. Drug repurposing is a hot topic and there are many published studies, but they are mostly limited to predictions for specific diseases or to non-translational algorithmic approaches. There exist also many biomedical databases which contain drug repurposing relevant information. However, these databases are scattered and not integrated into one unified database. Furthermore, various identifiers and syntax are used in available databases which need to be harmonized. As tempting as machine learning or deep learning approaches are for drug repurposing applications, they normally don't return interpretable results, and we need more mechanism-based approaches which return interpretable results by design. Algorithmic approaches are often fully automatic, and the involvement of expert knowledge which can enhance the accuracy is missing it. All being said, we need integrated and interactive tools which allow researchers from all biomedical disciplines to employ network-based drug repurposing approaches that are adaptable to their individual use cases. We began to address this need by COVAX, which allows the user to interact with the system, but is limited to COVID-19 drug repurposing. NeDRex generalizes and extends the approach implemented in COVAX to be applicable for other diseases. Unlike few other available tools, NeDRex doesn't confine users to a limited number of diseases and is applicable also to new diseases. Before introducing our platform, first we need to present the concept of disease module, which might be already known to many of you. Studies have indicated that disease-associated genes or proteins are not randomly scattered throughout biological networks. Instead, these genes and proteins tend to be located in so-called disease modules, which are small subnetworks representing interconnected mechanisms that can be linked to the phenotype. One of the bases of our network-based drug repurposing is that diseases can be viewed as perturbations of these disease modules. Consequently, potentially repurposable drugs can be identified in silico by carrying out the following 3 steps. First, a heterogeneous biological network is constructed by integrating data from multiple biomedical databases. This heterogeneous network contains knowledge that could be used for drug repurposing, for example, protein-protein interactions, disease gene, and drug-protein associations. Next, the constructed biological network is mined to derive disease modules associated with the disease of interest. Finally, drugs with known targets that are contained in or in close vicinity to the extracted disease modules are prioritized. An advantage of our platform is that users can engage their expert knowledge at 2 points where seeds need to be selected. The NeDRex platform has 3 main components: a knowledge base named NeDRex DB, a Cytoscape app named NeDRex app, and an API. The NeDRex app is a cytoscape app that provides implementations of network-based algorithms. These algorithms are made available to users via the RESTful API. The NeDRex DB integrates data from various biomedical databases, including OMIM, Drug Bank, and Uniport. Integration of multiple databases enables us to construct the heterogeneous networks described earlier. These networks can be accessed and explored via three means: the NeDRex app, the NeDRex API, and the Neo4j endpoint to the NeDRex DB. We integrated 10 different data sources covering genes, proteins, drugs, drug targets, disease annotations, and their relationships, resulting in an integrated network with 350,000 nodes and about 14 million edges. The metagraph shown here illustrates different types of integrated relationships between different node types, which are used for algorithms in the NeDRex platform and can be imported into NeDRex app. A number of the node and edge types are not included in the metagraph illustrated here, such as molecular similarity between drugs and associations representing proteins involved in pathways. However, users can access and download all of them via NeDRex API and use this data for their own repurposing methods. We have implemented multiple network-based algorithms in our platform for disease module identification and drug prioritization steps of the full drug repurposing pipeline. We implemented a modified version of standard closeness centrality to rank drugs. Here we compute the centrality with regarding to only the selected set of seed nodes and not all the nodes in the network. One of the NeDRex algorithms specifically tailored to the network medicine context is Multi-Steiner trees. The Steiner tree problem is a combinatorial optimization problem. It aims at finding the subgraph of minimum cost connecting a given set of seed nodes. For NeDRex, we established a multi-Steiner trees method that aggregates several approximates of Steiner trees into a single subnetwork. By selecting genes associated with a disease under study as seeds, MUST extracts a connected subnetwork which potentially incorporates the genes involved in the disease pathways and mechanism. The motivation behind returning multiple trees instead of one is that the solutions to the Steiner tree problem are usually non-unique, and computing several Steiner trees increases the stability of the extracted mechanism. Diamond is another disease module identification method which was originally developed by Rias and colleagues. Diamond identifies a candidate disease module around a set of known disease genes or seeds by greedily adding nodes with a high connectivity significance to the module. In this iterative algorithm, the connectivity significance of all direct neighbors of seeds is computed. Then the most significantly connected node is integrated into the module, leading to expansion of the module by one node per iteration. In contrast to MUST, Diamond does not necessarily return a connected subnetwork as the disease module. BICON, originally developed by Lazareva and colleagues, is another disease module identification algorithm implemented in our platform. BICON is a network constrained by clustering method that is used for integrative analysis of gene expression and PPI networks. It is an unsupervised approach that simultaneously performs patient and gene clustering such that the genes that provide the best possible clustering are also connected in a PPI network. Basically, BICON connects patient-specific information to prior knowledge about functional relationships. The retrieved subnetworks provide the direct explanation to the clustering and can be interpreted as disease modules. We implemented TrustRank for drug prioritization step. TrustRank ranks nodes in a network based on how well they're connected to a trusted set of nodes, which are seeds in our application. It is a diffusion-based prioritization method based on Google's PageRank algorithm, where trust is iteratively propagated from seed nodes to neighboring nodes using the network structure. The in silico network-based drug repurposing pipeline can be done in 3 main steps using the NeDRex app. The analysis begins by taking a set of genes as input. This gene set could be obtained by selecting the disease under study and subsequently obtaining genes associated with this disease from NeDRex. Alternatively, a custom set of genes can be provided, for example, differentially expressed genes associated with a disease of interest. In the next step, disease modules are derived using the seeds selected in step 1 by employing the must or diamond algorithms. Alternatively, the BICON algorithm can be employed to return disease modules. In this case, step 1 is skipped and gene expression data should be used as the input for this step. In the last step, drugs with targets in or in the vicinity of identified disease modules from previous steps are ranked. This last step can also be performed directly after step 1. As mentioned earlier, expert knowledge can be used to supplement the input to step 2 and step 3. To demonstrate the utility of the NeDRex platform to recover potential therapeutic drugs, I show you some example use case. Here, we are interested to identify repurposed drug candidates for inflammatory bowel disease. We start by selecting the IBD node and obtaining all genes associated with it from NeDRex-DB. Then we use these genes as input to MAS to identify the disease module. In total, 388 drugs target this module. Considering the high number of drugs targeting this module, we use the genes from the returned disease module as seeds for drug prioritization algorithms. 3 drugs among top rank ones have been indicated for the treatment of IBD, or the relevance to IBD are supported by literature evidence. None of these drugs target the initial disease seed genes. Fostamatinib was reported to alleviate IBD-induced inflammatory damage in rats. The JAK inhibitor ruxolitinib has been reported to ameliorate ulcerative colitis in a mouse model. Imatinib therapy has been reported to induce remission in IBD patients. Now an example of hypothesis-driven repurposing. We know that lipid and cholesterol metabolism has been linked with progression of Alzheimer's disease. We now want to investigate the shared mechanism between Alzheimer's and hyperlipidemia, and if drugs which are originally indicated for the treatment of hyperlipidemia appears in our results for repurposing for Alzheimer's. We start by identifying or obtaining genes associated to each disease and identifying their corresponding disease modules. We found 7 genes in common between the identified 2 disease modules. Notably, all of these genes are Alzheimer-associated genes, and some are well-characterized drivers of this disease. Importantly, most of the overlapping genes are not among the hyperlipidemia-associated genes, but they are retrieved only after obtaining the disease module with diamond. This demonstrates that in some cases, using only the disease-associated genes is not enough to uncover the molecular mechanisms shared between diseases, and using the disease module provides actually a more complete landscape of the disease. By prioritizing drugs using this intersection set as seeds, we found gemfibrozil among the top-ranked drugs. This drug indirectly targets the intersection set. Gemfibrozil is originally indicated for the treatment of hyperlipidemia and is being tested in clinical trials, and preclinical studies give evidence of potential effectiveness of this drug for the treatment of Alzheimer's. I invite you to try out our tool, which is available at NeDRex.net. There you can download the NeDRex app and also find tutorial videos on how to use the app with some real-world examples. To summarize, we developed a flexible and interactive tool for a full analysis pipeline that integrates multiple network-based algorithms to identify potential targets and drug candidates for drug repurposing. Our tool benefits from expert-in-the-loop paradigm. It retains interpretable results, and it allows for the visualization of the results as networks in Cytoscape app, which allows exploring the results further by additional functions available in the app. Finally, I would like to acknowledge the co-authors for their valuable contribution to this project, especially James Skelton. Also a big thank to my supervisors, Dr. Blumenthal, Dr. Kaprowski, and Dr. Bambach for their great support and advice during this project. I'd also like to thank EU for funding this project. And last of all, thank you all for listening.